Every atom in your body is replaced, every single one. Studies at Oak Ridge National Laboratory showed that 98% of them are replaced every year. You get a new suit of skin every month, a new liver every 6 weeks, a new skeleton every 10 years. In a decade, nothing that is part of you today will still be there. So why will you be older? If every piece of you is new, fresh atoms just arrived, never been part of you before, why does your body not feel new? Why do your joints ache? Why does your skin wrinkle? Why do you forget things you used to remember? The carbon in your muscles, the calcium in your bones, the iron in your blood, all of it is swapped out for new atoms drawn from the food you eat, the water you drink, the air you breathe. The answer involves the second law of thermodynamics, quantum mechanics, nuclear physics, and a thought experiment from 1867 that describes exactly what is happening inside every one of your cells. The speed of this replacement varies enormously depending on the chemical environment. The water in your body, and you're roughly 60% water, is exchanged every 16 days. That means 3 weeks from now, almost none of the water molecules currently inside you will still be there. They will have been exhaled, sweated out, excreted, and replaced by new water molecules from your next few hundred glasses of water. Red blood cells last about 120 days. The cells that line your intestines are replaced every 3 to 5 days. They live in an environment so harsh, so acidic, so chemically violent that they are essentially disposable. Your body manufactures them, uses them, destroys them, and manufactures replacements over and over, thousands of times across your lifetime. But some atoms stay longer than others. The carbon atoms in the structural proteins of your tendons and cartilage can persist for decades. Some neurons in your brain and some cells in your heart muscle are never replaced. They are with you from birth to death. The atoms in your tooth enamel, once laid down in childhood, remain essentially unchanged for the rest of your life. So the replacement is not uniform. Most of you is constantly churning. A few critical parts are there for the entire ride. And here's a number that puts this into perspective. Every single breath you take exchanges roughly 2 times 10 to the 21st atoms with your environment. 2 sextillion atoms, in and out, per breath. You take about 20,000 breaths per day. The sheer volume of atomic traffic through your body is staggering. You are not a fixed object. You are a river of atoms flowing through a shape. You are not the same physical object you were a decade ago. The atoms are completely new, and they are fine. A carbon atom is a carbon atom whether it is in a newborn or a 90-year-old. The atoms do not age. Atoms do not wear out. A hydrogen atom in your body right now is the same hydrogen atom it was 13.8 billion years ago when it was formed in the first minutes after the Big Bang. It has been inside stars. It has been part of interstellar gas clouds. It has been in oceans and organisms and rocks, and now it is in you, and it is exactly the same as it ever was. The atoms are not the problem, so what is? Think about it this way. Imagine building a sandcastle. 4 walls, turrets, a central spire, a moat. Beautiful structure. Now start replacing 100% of the sand with brand new sand. Grain by grain, swap every piece of the old castle for fresh material. What happens to the structure? The walls start to sag. The turrets lose their shape. The moat fills in. Not because the new sand is worse than the old sand. The sand is identical. The problem is that during the replacement, the structure degrades. The information, the precise arrangement of grains that made the castle look like a castle, is lost little by little with every swap. That is what is happening to your body. You are not your atoms. You cannot be, because your atoms change constantly. You are the pattern. You are the arrangement. You are the information that tells 7 octillion atoms where to go and what to do and how to organize themselves into a structure so complex that it can walk and talk and wonder why it is getting old. But the real question is, what is destroying the pattern? The answer involves something happening inside you right now that you cannot feel. This is a very old philosophical problem dressed in modern physics. The Greeks called it the Ship of Theseus. If you replace every plank of a ship one at a time until no original material remains, is it still the same ship? Applied to your body, the answer is both yes and no. You are the same person in terms of continuity. The pattern was maintained through each replacement. But you are not the same object in terms of material. You are a pattern moving through matter. A wave, not a particle. Think about a wave in the ocean. The water molecules in a wave are constantly changing. New water enters the wave at the front. Old water leaves at the back. The wave moves across the surface of the ocean, but no single molecule of water travels with it. The wave is not a thing. It is a pattern, a shape maintained in a medium. You are the same kind of phenomenon. A pattern maintained in atoms. A temporary, self-sustaining arrangement of matter that persists for decades because the information that defines the arrangement is continuously copied and enforced by molecular machinery inside every one of your 37 trillion cells. Or think about a candle flame. The flame has a shape, a color, a temperature. It persists for hours, but no single molecule of gas stays in the flame for more than a fraction of a second. Wax vapor rises, ignites, burns, and is replaced by new wax vapor rising behind it. The flame is not an object, it is a process, a continuous chemical reaction that maintains a stable pattern in a flow of matter and energy. You are closer to a flame than you are to a rock. You are a process, not a thing. In the language of information theory, the framework developed by Claude Shannon in 1948, you are information instantiated in matter. Your DNA is a digital code written in an alphabet of 4 letters, storing the instructions for building and maintaining the pattern. Your genome contains roughly 750 megabytes of data. That is less than a modern smartphone app. But that 750 megabytes, read and executed by the molecular machinery of your cells, is enough to organize 7 octillion atoms into a conscious being. A computational neuroscientist named Terence Sejnowski put it this way. He said he has been puzzled by his ability to remember his childhood even though most of the molecules in his body, including the molecules that make up his brain, have been replaced many times over. The memories persist. The molecular substrate does not. The information outlasts the material. But here is the thing about patterns. They require information to persist, and no copying process in the physical universe is perfect. This is not a limitation of biology, it is a limitation of physics. The third law of thermodynamics implies that perfect copying would require infinite energy. Every physical copying process, whether it is a Xerox machine, a vinyl record press, or a DNA polymerase, introduces noise. And noise is information loss. This is where the physics of aging begins. Your DNA is the master copy of the information. It is the blueprint. 3.2 billion base pairs, encoding roughly 20,000 genes, packed into the nucleus of nearly every cell in your body. If you stretch the DNA from a single cell into a line, it would be about 2 meters long. If you stretch the DNA from all 37 trillion cells in your body, it would reach from the Earth to the Sun and back more than 300 times. That is roughly 100 billion kilometers of DNA inside you right now. All of it encoding the same fundamental instruction set, all of it being copied constantly. Every time a cell divides, the entire sequence must be copied. All 3.2 billion base pairs, replicated with extraordinary precision. The molecular machinery that does the copying, a complex of enzymes centered on DNA polymerase, is the most accurate copying system known to exist anywhere in nature. It reads the template strand, selects the correct complementary nucleotide from the surrounding solution, catalyzes the bond, and moves to the next position. It does this at a rate of roughly 1,000 base pairs per second. To copy the full genome, multiple polymerase complexes work simultaneously at different points along the chromosomes. The entire 3.2 billion base pair genome is copied in roughly 8 hours. The raw error rate before proofreading is roughly 1 mistake per 10,000 to 100,000 base pairs. That sounds terrible, but the system has layers of error correction that are elegant in their engineering. DNA polymerase has a built-in proofreading function, a separate catalytic site that detects mismatched base pairs immediately after they are placed and removes them. After that, a mismatch repair system scans the newly copied strand and corrects errors that the proofreading missed. There are also base excision repair and nucleotide excision repair pathways that fix damage caused by environmental factors, ultraviolet radiation, reactive chemicals, background ionizing radiation. Your body is running multiple overlapping error correction systems simultaneously all the time in every cell. After all corrections, the final error rate is roughly one mistake per billion base pairs per replication, 1 in a billion. That is a copying fidelity of 99.9999999%. If a human typist achieved this accuracy, they could type the entire Encyclopedia Britannica 3 times before making a single error. It is the most precise information transfer system in the known universe. It is not perfect. And copying errors are not even the main problem. Your DNA is under constant assault from physics itself. You are radioactive. Right now, sitting wherever you are, roughly 4,400 atoms of potassium-40 are decaying inside your body every second. Potassium is essential for nerve function and muscle contraction. Your body maintains about 140 grams of it at all times. A tiny fraction, roughly 0.012%, is the radioactive isotope potassium-40, which has a half-life of 1.25 billion years. It has been decaying since before the Earth formed, and it decays inside your cells, emitting beta particles, high-energy electrons, that slam into your DNA at roughly 80% the speed of light. Each impact can break a strand, damage a base, or cause a mutation. Your own atoms are irradiating your own genome. This is not an external threat, it is built into your chemistry. That is bad enough. But the assault is not just coming from inside your body. Cosmic rays, high-energy particles originating from supernovae, black holes, and other violent astrophysical events, rain down on the Earth continuously. Most are absorbed by the atmosphere, but secondary particles called muons reach the surface. Roughly 10,000 muons pass through every square meter of the Earth's surface every minute. 1 muon per second per square centimeter passes through your body. Each muon has enough energy to ionize atoms along its path, breaking chemical bonds, including the bonds that hold your DNA together. You cannot feel this, you will never notice it. But it is happening right now, inside every cell you have. And there is one more source of mutation, stranger than all the others, one that operates at the boundary of physics itself. Quantum tunneling. The base pairs in your DNA are held together by hydrogen bonds. The protons in those bonds have a small but non-zero probability of quantum tunneling to the wrong position, jumping from one side of the bond to the other without passing through the energy barrier in between. This was first theorized by Per Olav Ludin in 1963. He proposed that if a proton tunnels to the wrong position, it changes the shape of the base pair, and when the DNA is copied, the polymerase reads the wrong letter. A quantum mechanical mutation. In 2022, computational studies using quantum dynamics simulations confirmed that proton tunneling occurs in DNA at biologically relevant rates. Aging has a quantum mechanical component. Some of the errors in your genome are not chemical accidents. They are quantum events governed by the same probability rules that govern radioactive decay and electron behavior. Between the copying errors, the radioactive decay, the cosmic ray hits, and the quantum tunneling events, your DNA sustains roughly 10,000 to 100,000 individual lesions per cell per day. 10,000 to 100,000 per cell per day. And you have 37 trillion cells. Your repair systems handle the vast majority of this damage. They are extraordinary, overlapping layers of molecular machinery that proofread, scan, snip, patch, and stitch broken DNA back together, running in every cell all the time around the clock. They catch almost everything, but they do not catch everything, and the fraction they miss accumulates. One error per billion across 3.2 billion base pairs means roughly 3 to 4 errors per cell division. Your body produces approximately 3.8 million new cells per second, every hour, every day of your life. That is 3.8 million copying events, each introducing a small number of errors each second. Over the course of a single year, your cells will have undergone roughly 10 to the 16th individual base pair replications. The numbers are so large they lose meaning, but the errors are real and they are cumulative. Most of these errors are harmless. The human genome is roughly 98% non-coding, regions that do not directly produce proteins. An error in a non-coding region usually has no effect, and even errors in coding regions often produce changes so minor that the resulting protein functions normally. A single amino acid substitution in a protein that is 300 amino acids long may change nothing about how that protein folds or what it does. But some errors hit critical targets. Tumor suppressor genes, the genes that prevent cells from dividing uncontrollably. DNA repair enzymes, the very proteins whose job it is to fix copying errors. Cell cycle regulators, apoptosis pathways, the systems that tell damaged cells to self-destruct for the good of the organism. When these genes are damaged, the consequences compound. A damaged repair gene means more errors in the next round of copying. More errors mean a higher chance of hitting another repair gene. The system degrades its own ability to prevent further degradation. This is not a stable equilibrium. It is a positive feedback loop, a cascade. But copying errors are only half of the clock. There is a second countdown running in every cell, and unlike mutations, this one is not random. It is precise, it is mechanical, and it is irreversible. The ends of your chromosomes are capped with structures called telomeres. These are repetitive sequences of DNA, the same six-letter code, TTAGG, repeated roughly 2,000 times at each chromosome end. Telomeres do not code for anything. They carry no instructions. They are buffers. Protective caps. Their function is to prevent the chromosome from unraveling or fusing with neighboring chromosomes, the way the plastic tip on a shoelace, an aglet, prevents the fabric from fraying. Every time a cell divides, the telomeres get slightly shorter. This is not a biological accident. It is a consequence of the geometry of DNA replication, a problem rooted in the physical structure of the molecule. DNA polymerase synthesizes new strands in only one direction, from the 5' end to the 3' end. On one strand, the leading strand, this works fine. On the other strand, the lagging strand, the polymerase must work in short segments called Okazaki fragments, and it cannot initiate synthesis at the very tip of the chromosome. There is always a small stretch at the end that cannot be copied. This is the end replication problem, first described theoretically by the Russian biologist Alexei Olovnikov in 1971, and it means that with every cell division, your chromosomes lose roughly 50 to 200 base pairs of telomeric DNA from each end. 50 to 200 base pairs per division, per chromosome end. It does not sound like much, but over 40, 50, 60 years of cell divisions, it adds up. When the telomeres get too short, the cell receives a signal to stop dividing permanently. This is the Hayflick limit, named after Leonard Hayflick, who discovered it in 1961. He observed that human cells in culture could divide roughly 40 to 60 times before they stopped. Nobody believed him at first. The prevailing view was that normal cells were immortal in culture. Hayflick proved they were not. There was a built-in counter, and the counter was the telomere. After reaching the Hayflick limit, the cell enters a state called senescence. It is alive, but it is no longer dividing. It is no longer contributing to tissue renewal, and it is often actively harmful. Senescent cells secrete a cocktail of inflammatory molecules called the senescence-associated secretory phenotype, interleukins, chemokines, matrix metalloproteinases. These signals damage surrounding healthy tissue, promote chronic inflammation, and can even push neighboring cells towards senescence. One damaged cell makes its neighbors worse. Another cascade. Telomere length correlates inversely with biological age. This has been measured across species, across populations, across individuals. Longer telomeres, younger biology; shorter telomeres, older. The longest-lived organisms tend to have longer telomeres and greater resistance to stress-induced telomere shortening. The connection is so reliable that telomere length is used as a biomarker of aging in clinical research. Not a perfect biomarker. Lifestyle, genetics, and stress all modulate the rate, but a consistent one. Something remarkable was observed during NASA's twin study. Astronaut Scott Kelly spent 340 days on the International Space Station while his identical twin brother Mark remained on Earth. During the mission, Scott's telomeres lengthened, an unexpected result possibly related to increased telomerase activity in the space environment, or changes in cell division rates under microgravity, or altered gene expression triggered by cosmic radiation exposure. Nobody is entirely sure of the mechanism. But when Scott returned to Earth, his telomeres rapidly shortened again, overshooting their pre-flight length and ending up shorter than when he started. The space environment temporarily reversed one aspect of the aging clock. The return to normal gravity made it snap forward. The pattern was disrupted and the pattern fought back, but not in his favor. Now, there is an enzyme that can rebuild telomeres. It is called telomerase, and its function is straightforward. It adds new TTAGGDGG trays to chromosome ends, compensating for the losses incurred during replication. If your cells express telomerase continuously, your telomeres would never shorten and your cells could, in principle, divide indefinitely. But most adult human cells have telomerase switched off. The gene is silenced, the enzyme is not produced, and there is a very good reason for that. Cancer cells reactivate telomerase. In roughly 85 to 90% of human cancers, telomerase is turned back on. That is one of the key steps in the transformation of a normal cell into a cancer cell. By reactivating telomerase, the cancer cell escapes the Hayflick limit. It can divide without end. It becomes effectively immortal at the cellular level. This is the dark irony of aging. The mechanism that could theoretically prevent cellular aging is the same mechanism cancer uses to become unkillable. The body faces a fundamental evolutionary trade-off: allow unlimited cell division and risk cancer, or limit cell division and guarantee aging. Evolution chose aging. The logic is straightforward. Cancer can kill you before you reproduce, aging kills you after. But DNA errors and telomere shortening are specific mechanisms. They are the how. The deeper question is why. Why does information degrade? Why do copies get worse? Why does the pattern inevitably blur? The answer is the most fundamental law in physics, and it means aging is not a biological problem, it is a physics problem. And physics problems do not have biological solutions. To understand what the second law has to do with your body, you need to understand what entropy actually measures. It is not just disorder, that is the popular shorthand, but it misses the precision. Entropy, as defined by Ludwig Boltzmann in 1877, is proportional to the logarithm of the number of microscopic arrangements that correspond to a given macroscopic state. His equation, S equals K log W, is carved on his tombstone in Vienna. It is one of the most important equations in the history of physics. What it means is this: a system has low entropy when very few microscopic arrangements produce the observed macroscopic state. A neatly sorted deck of cards has low entropy. There is exactly one way to arrange 52 cards in perfect order from Ace of Spades through King of Clubs. A shuffled deck has high entropy. There are roughly 8 times 10 to the 67th possible disordered arrangements. Almost any random arrangement is a disordered one. Order is rare. Disorder is overwhelmingly probable. The second law says that in any spontaneous process, the total entropy of a closed system never decreases. Things move from rare arrangements to probable ones, from order to disorder. Shuffled decks do not spontaneously sort themselves. Broken glasses do not reassemble. Hot coffee does not spontaneously get hotter while the room gets colder. Heat flows from hot objects to cold objects, never the reverse. Your body is, by any measure, one of the most ordered structures that matter has ever been arranged into on this planet. 7 octillion atoms. That is a 7 followed by 27 zeros. Precisely organized into molecules, organized into organelles, organized into cells, organized into tissues, organized into organs, organized into systems, all coordinated to maintain a living, thinking, conscious being. The entropy of this arrangement is spectacularly low. The number of ways to arrange 7 octillion atoms into a functioning human body is vanishingly, almost immeasurably small compared to the number of ways to arrange them into anything else. A pile of dust, a puddle of water, a cloud of gas. You are thermodynamically improbable. By the logic of the second law, you should not exist. And yet you do, because of a trick. The trick is energy. Your body is not a closed system. It is an open system. It exchanges matter and energy with its environment continuously, and this changes everything. The second law says the total entropy of a closed system never decreases, but a local region within an open system can decrease its entropy, can become more ordered, as long as it exports enough entropy to its surroundings to more than compensate. This is what you do every second of your life. You eat low-entropy food, structured molecules with chemical energy stored in their bonds. You break those molecules apart, extract the energy, use it to maintain your internal order, and export the waste as high-entropy heat and carbon dioxide and urea. You are an entropy pump. You take in order and excrete disorder. In 1944, the physicist Erwin Schrödinger wrote a book called What is Life?, in which he asked how living organisms maintain their extraordinary order in a universe that tends relentlessly toward disorder. His answer: a living organism feeds on negative entropy. It drinks orderliness from its environment. That phrase, negative entropy or negentropy, became one of the foundational concepts of biophysics. Life, in Schrödinger's framework, is not a violation of the second law. It is a local, temporary reversal purchased at the cost of greater entropy increase elsewhere. The sun radiates low-entropy photons. Plants capture them. You eat the plants. The total entropy of the Sun-Earth system increases. But inside your body, for a while, order is maintained. But here is the critical point. The point that explains aging at the deepest level of physics. Every mechanism your body uses to fight entropy is itself made of molecules, and those molecules are subject to the same thermodynamic pressures as everything else in the universe. There is a thought experiment in physics that describes your situation exactly, not approximately, not metaphorically, exactly. It was proposed in 1867, and it took over a century to resolve. And when it was resolved, it proved something that applies directly to every cell in your body. James Clerk Maxwell imagined a tiny intelligent being, later called Maxwell's demon, stationed at a door between two chambers of gas. The demon watches molecules approach the door and selectively opens it to let fast molecules through one way and slow molecules through the other. Over time, one chamber gets hot and the other gets cold, apparently violating the second law of thermodynamics. Order from disorder. Entropy decreasing without any work being done. For over a century, physicists debated whether Maxwell's demon could actually work. In 1961, Rolf Landauer proved that it could not. Landauer's principle states that erasing information, which the demon must do to reset its memory after each decision, always dissipates a minimum amount of energy as heat. The demon creates order, but the act of creating order requires information processing, and information processing produces entropy. The total entropy of the system still increases. The demon cannot win. It can only slow the rate of loss. Your repair enzymes are Maxwell's demons. Every DNA repair protein, every chaperone that refolds a misfolded protein, every antioxidant that neutralizes a free radical, they are all doing what Maxwell's demon does. They are sorting, selecting, maintaining order against the tide of disorder, and every one of them pays the Landauer cost. Every repair event requires ATP. Every quality control check consumes energy. The demons run on fuel, and when the fuel supply drops, when mitochondrial function declines, when ATP production falls, the demons slow down. They miss more errors. They let more damage through. The sorting becomes less precise. The order degrades faster. The DNA that encodes your repair proteins accumulates copying errors. The proteins that repair your DNA are produced by DNA that is itself accumulating errors. The mitochondria that produce the energy required for all of this repair work are themselves damaged by the byproducts of energy production. The repair systems need repair, the maintenance systems need maintenance, and the systems that maintain the maintenance systems also need maintenance. It is recursive. There is no bottom. There is no level at which the machinery is exempt from the laws of physics. The mathematician Manfred Eigen described a related concept in molecular evolution called the error catastrophe threshold. There is a critical mutation rate above which the information content of a genome cannot be maintained across generations. Below the threshold, selection can purge errors faster than they accumulate. Above it, the genome disintegrates, the information dissolves into noise. Your body's cells face an analogous problem. As long as the repair rate exceeds the damage rate, the cellular genome remains stable. But as repair capacity declines with age and damage rate remains constant or increases, individual cell lineages approach their own error catastrophe. The information in the DNA of an 80-year-old cell is measurably noisier than the information in a 20-year-old cell. The genome is not failing catastrophically. Not yet, but it is drifting toward the threshold. This is why aging is not a disease. It is not a malfunction. It is not something that went wrong. It is the second law of thermodynamics expressed in biology. The repair systems, your Maxwell's demons, hold off entropy for decades. That is an astonishing achievement, the most impressive thermodynamic trick in the known universe. But they cannot hold it off forever, because they are made of the same stuff they are trying to protect. They are molecules fighting the degradation of molecules. They are entropy trying to fight entropy. And entropy always wins that fight. The physicist Ilya Prigogine, who won the Nobel Prize for his work on thermodynamic systems far from equilibrium, described living organisms as dissipative structures. You are a far from equilibrium system that maintains its internal order by continuously dissipating energy into the environment. You are a temporary eddy of order in a river of increasing entropy. The eddy persists as long as the energy flows through it and the structures that channel the energy remain intact. When the energy flow weakens or when those structures degrade, the eddy dissipates. The river wins. And evolution does not oppose this. It does not even try. Natural selection optimizes for reproductive success, not longevity. Your repair systems are good enough to get you to reproductive age and keep you functional long enough to raise offspring to independence. After that, the selection pressure drops dramatically. In 1957, the evolutionary biologist George Williams formalized this as antagonistic pleiotropy. Genes that boost your reproductive fitness at age 20 can actively harm you at age 70, and natural selection will not remove them because by 70 your genes have already been passed on. The harm comes too late for evolution to care about. Your body was never designed to last. It was designed to reproduce. Lasting was never the objective, so the pattern degrades, the information decays, the entropy increases, and the atoms, the perfectly fine, perfectly new, perfectly ageless atoms, assemble into a slightly worse copy of you, year after year, decade after decade, until the accumulated degradation overwhelms the repair systems and the pattern can no longer sustain itself. That is aging. Not wear and tear, not time passing. It is the slow, inevitable loss of information, the pattern that is you, under the pressure of the most fundamental law in physics. Your repair systems hold the line for decades. Your Maxwell's demons sort and fix and rebuild with a precision that no human technology has ever matched. They are the most extraordinary molecular machinery in the known universe. But they are made of the same stuff they are trying to protect, and that is why the fight always ends the same way. But how it ends, the specific chain of failures, the way entropy expresses itself inside your tissues and organs and bones, is something you can actually see, something you can measure. And it starts with the engines that keep every cell alive. and the damaging waste they produce as a byproduct of doing their job. The engines are called mitochondria. There are 10 million billion of them inside you, and they are poisoning you right now, not as a malfunction, but as a requirement of the chemistry that keeps you alive. To understand why, you need to understand the scale of what they do. Your body produces its own weight in fuel every single day, not food, fuel. The molecular fuel that powers every process in every cell, a molecule called adenosine triphosphate, ATP. You carry only about 50 grams of it at any given moment, but you burn through it so fast that your body manufactures and recycles roughly 40 to 70 kilograms of it every 24 hours. That is your own body weight in chemical energy, synthesized from scratch, daily. Every muscle contraction, every nerve impulse, every protein being built, every strand of DNA being copied, every ion being pumped across a membrane, all of it runs on ATP. Without it, you are dead in seconds, not minutes, seconds. These factories are tiny, roughly 1 to 10 micrometers long, and there are approximately 10 million billion of them in your body. That is, 10 to the 16th power. Every cell contains hundreds to thousands of them, packed in wherever energy demand is highest. Heart muscle cells contain roughly 5,000 mitochondria each, because the heart never stops beating and never stops demanding fuel. Mitochondria do something remarkable. They take the chemical energy stored in the food you eat, glucose, fatty acids, and combine it with the oxygen you breathe to produce ATP. The process is called oxidative phosphorylation, and it takes place across a membrane inside the mitochondrion, where a chain of protein complexes passes electrons from one to the next, each transfer releasing a small amount of energy that is used to pump protons across the membrane. The protons build up on one side, creating an electrochemical gradient, essentially a tiny battery. Then they flow back through a molecular turbine called ATP synthase, which literally spins like a rotary engine and uses the energy of the proton flow to attach a phosphate group to ADP, converting it to ATP. ATP synthase rotates at roughly 9,000 revolutions per minute. It is a nanoscale machine with moving parts, a rotor, a stator, a central axle, operating on the same mechanical principles as a hydroelectric turbine. The mechanism that drives it, proposed by Peter Mitchell in 1961 and ridiculed for nearly 2 decades before earning him the Nobel Prize, is called chemiosmotic coupling. The proton gradient across the membrane is not just a chemical phenomenon. It is an electrical one. The physics of this process is electrochemistry. The voltage across the inner mitochondrial membrane is roughly 180 millivolts. That sounds small, but the membrane is only 5 nanometers thick. Divide the voltage by the thickness, and you get a field strength of roughly 30 million volts per meter. That is in the same range as a lightning bolt. Every mitochondrion in your body maintains a lightning bolt strength. Electric field across its inner membrane all day, every day, for your entire life. If that field collapses, if the membrane is breached and the proton gradient dissipates, ATP production stops and the cell dies. But there is a cost, and it is not a side effect, it is not a malfunction. It is built into the chemistry of the fuel itself. The very molecule that keeps you alive is also the molecule that is destroying you. You breathe oxygen to survive. But oxygen is a poison. This is the fundamental paradox of aerobic life. Oxygen is the terminal electron acceptor in the electron transport chain, the molecule that receives the electrons at the end of the relay and combines with protons to form water. Without oxygen, the chain backs up, the proton gradient cannot be maintained, and ATP synthesis stops. You need oxygen to live. But oxygen is one of the most reactive elements in the periodic table. It wants electrons. It takes them from whatever is nearby. And when it takes them from the wrong molecule, the result is destruction. The electron transport chain is not perfectly efficient. Roughly 1 to 2% of the electrons leak out at complexes 1 and 3 and react directly with oxygen to form superoxide, a reactive oxygen species, a free radical, an oxygen molecule with an unpaired electron which makes it chemically desperate. It will rip electrons from whatever molecule is nearest: a lipid in the membrane, an amino acid in a protein, a base in a strand of DNA. The damage is instant and indiscriminate. This is oxidation, the same chemical process that rusts iron, browns a cut apple, and degrades rubber. Your body is rusting from the inside, not as a metaphor, as chemistry. The same reaction, the transfer of electrons from one substance to oxygen, is happening inside your cells continuously as a direct byproduct of the process that keeps you alive. The thing that gives you energy is also the thing that damages you. You cannot have one without the other. This is not a design flaw. It is thermodynamics. Your body produces antioxidant defenses: superoxide dismutase, catalase, glutathione peroxidase, enzymes that neutralize free radicals before they can do damage. In a young body, these defenses are robust. They catch most of the leaked electrons and prevent most of the damage. But the defenses are proteins. Proteins are encoded by DNA. DNA accumulates errors. The defenses weaken with age, just as the rate of free radical production remains constant or increases. The gap between damage and defense widens every year. And the mitochondria themselves are the most vulnerable targets. They contain their own DNA, a small circular genome of roughly 16,500 base pairs, encoding 37 genes, many of which are essential for the electron transport chain. Mitochondrial DNA sits right next to the electron transport chain, unprotected by the histone proteins that shield nuclear DNA. It has fewer repair mechanisms. It accumulates mutations 5 to 10 times faster than nuclear DNA. Damaged mitochondrial DNA produces damaged electron transport chain components. Damaged components leak more electrons. More leaked electrons produce more free radicals. More free radicals damage more mitochondrial DNA. This is a vicious cycle, a degradation spiral with no external cause. The system poisons itself. ATP production declines roughly 8% per decade after age 30. By 70, your cells are producing approximately 30% less energy than they did at their peak. Less energy means less fuel for the Maxwell's demons: the repair enzymes, the chaperones, the quality control systems. The demons slow down. They miss more. The pattern blurs faster. And the damage is not limited to the inside of the cell. It is written on your body in a way you can see in the mirror. Because the same chemical principles that rust iron inside your mitochondria are also caramelizing the structural proteins that hold you together. Collagen is the most abundant protein in the human body. It makes up roughly 30% of all your protein. It is the structural scaffold, the load-bearing framework of your skin, your bones, your tendons, your blood vessels, your cartilage, your corneas, your gut wall. If you removed everything from your body except the collagen, you would still be recognizable. It is the architecture. Collagen is also one of the longest-lived proteins in your body. In bone, collagen turns over every 1 to 2 years. In skin, every 10 to 15 years. In cartilage, the flexible tissue in your joints, your nose, your ears, the collagen lasts over 100 years. Some of the collagen in your knee cartilage right now was manufactured when you were a child. It has been there your entire life. And that long lifespan is precisely what makes it vulnerable. There is a chemical reaction called glycation. It is the non-enzymatic reaction between a sugar, typically glucose, and a protein. Glucose molecules floating in your blood and interstitial fluid randomly collide with proteins and form bonds that, over months and years, become permanent. The end results are called advanced glycation end products, AGEs. Some AGEs are adducts, they modify a single protein. But others are crosslinks. They form permanent chemical bridges between two neighboring protein molecules. The most abundant cross-linking AGE in collagen is glucosapine, a lysine-arginine bridge. Once formed, AGE crosslinks are virtually irreversible. The body has no enzyme that can break them. They are permanent modifications that accumulate over your entire lifetime. The chemistry will sound familiar. It is the Maillard reaction. The same reaction that browns bread when you toast it caramelizes the surface of a steak when you sear it, turns onions golden when you cook them slowly. It is a sugar reacting with a protein under heat. In cooking, the heat is external and the reaction takes minutes. In your body, the heat is your body temperature, 37 degrees Celsius, and the reaction takes years. But it is the same chemistry. Your body is slowly cooking itself. The browning of your tissues is the same process as the browning of a steak, just immeasurably slower. The effects are visible and measurable. Cross-linked collagen in the skin loses its elasticity. The skin cannot stretch and rebound the way it used to. Wrinkles form. Cross-linked collagen in blood vessel walls makes the vessels stiffer. They cannot absorb the pressure wave of each heartbeat as efficiently. Blood pressure rises. The heart works harder. Cross-linked collagen in the lens of the eye makes the lens opaque. Light scatters instead of passing through cleanly. This is cataract. The same process stiffens the cartilage in your joints, the filtration membranes in your kidneys, the elastic tissue in your lungs. Every tissue that depends on collagen for its mechanical properties is degraded by this reaction. And the process accelerates with blood sugar. People with type 2 diabetes who have chronically elevated blood glucose accumulate AGEs much faster. Their tissues stiffen sooner. Their wrinkles are finer and more numerous. Their skin sometimes takes on a yellowish hue, the visible color of advanced glycation, the same golden brown as the Maillard reaction in cooking. Some researchers call type 2 diabetes a premature aging disease. The physics supports that description. It is the same process, running at a higher rate. Pentosidine, one specific AGE crosslink, increases roughly 200% in skeletal muscle collagen between age 25 and age 78. Your collagen at 78 is chemically different from your collagen at 25. Not because the collagen is old, some of it is, but much of it has been replaced. The crosslinks are the problem. They accumulate on new collagen too, as soon as it is manufactured. The pattern is compromised not just in the information, but in the physical material that embodies it. But aging does not only stiffen your structures, it corrupts the machines that build them. And the way it does this involves one of the deepest problems in molecular physics, a problem so fundamental that it explains why your brain produces the plaques and tangles of Alzheimer's disease. Your body manufactures roughly 20,000 different types of proteins. Each one is a chain of amino acids, anywhere from a few dozen to several thousand, that must fold into a precise three-dimensional shape to function. A protein's function is entirely determined by its shape. An enzyme's active site must be the exact right geometry to catalyze its specific reaction. A receptor on a cell surface must have the exact right shape to bind its specific signal molecule. A structural protein must form the exact right fibers to bear the loads it is designed for. If the shape is wrong, the function is lost. There is no partial credit in protein folding. A protein that is 99% correctly folded can be completely non-functional. In 1972, Christian Anfinsen won the Nobel Prize for demonstrating that the information needed to fold a protein is contained entirely in its amino acid sequence. The sequence determines the shape. Any corruption of the instructions can produce a protein that folds wrong, and the folding itself is almost miraculous. A protein with just 100 amino acids has more possible configurations than there are atoms in the observable universe. If it searched randomly, finding the right shape would take longer than the age of the universe. It finds it in milliseconds, guided by thermodynamic forces that funnel it toward the correct fold, like a ball rolling to the bottom of a valley in an energy landscape. But the correctly folded state is not the most stable state. The absolute lowest energy state for most proteins is an amyloid fibril, a long, rigid, highly ordered aggregate of many protein copies stacked together. Amyloid fibrils are thermodynamically more stable than correctly folded proteins. The native fold is a kinetic trap, held in place by energy barriers, not the thermodynamic ground state. Given enough time, enough perturbation, enough accumulated damage, proteins tend toward amyloid. This is the deep physics of why aging produces plaques and tangles. The native fold is a temporary victory over thermodynamics. Amyloid is where the physics wants the protein to end up. The correctly folded state is a low entropy state. It is one specific arrangement out of an astronomical number of possible arrangements. Maintaining that state requires energy and the help of chaperone proteins. Molecular machines that assist folding detect misfolded proteins and either refold them or tag them for destruction by the cell's waste disposal system, the proteasome. As you age, chaperone function declines. The proteasome becomes less efficient, and misfolded proteins accumulate. They clump together into aggregates, disordered tangles, and plaques that the cell cannot remove. These aggregates are the physical basis of some of the most devastating age-related diseases. In Alzheimer's disease, beta-amyloid proteins misfold and aggregate into plaques between neurons, while tau proteins misfold and form tangles inside neurons. In Parkinson's disease, alpha-synuclein misfolds and forms Lewy bodies in dopamine-producing neurons. In both cases, the disease is a protein folding failure. Entropy winning at the molecular level, the low entropy folded state collapsing into high entropy disorder. And the connection to glycation makes it worse. Glycated proteins, proteins modified by the same sugar cross-linking that stiffens your collagen, are more prone to misfolding and aggregation. Glycated beta-amyloid aggregates faster. Studies have found approximately 3 times higher levels of AGE, modified proteins in the brain plaques of Alzheimer's patients compared to healthy age-matched controls. The Maillard reaction is not just stiffening your tissues, it is promoting the protein aggregation that destroys your brain. Every one of these processes is a specific expression of the same underlying physics. Entropy, the second law. The degradation of information and structure under the relentless pressure of thermodynamic probability. But there are 2 more changes that are easy to miss, not because they are subtle, but because they are so slow that you only notice them in decades. And one of them, statistically, may be more dangerous than everything else in this video combined. You are drying out. The average person loses 1 to 2 inches of height between ages 30 and 70. The reason is water. A newborn is 75% water. By the time you are elderly, you are closer to 50%. You have lost a quarter of the substance that makes up most of you, and every cell in your body feels the difference. Water is not passive filler. It is the medium in which all biochemistry occurs, the solvent that makes every reaction possible. Water molecules form a dynamic shell around every protein, every DNA strand, every membrane in your body. This hydration shell is structural. Proteins do not fold correctly without it. Enzymes do not function without it. The loss is gradual and systemic. Intracellular water decreases. Your cells literally shrink with age. The intervertebral discs in your spine, water-filled cushions between your vertebrae, dehydrate and compress. Skin loses water content as hyaluronic acid declines. A single molecule of hyaluronic acid can hold up to 1,000 times its weight in water, and as levels drop, the skin loses volume and resilience. Meanwhile, the kidneys become less efficient at concentrating urine, meaning you lose more water per day, while your thirst response becomes less sensitive, meaning you drink less. The system loses water faster and replaces it slower. The gap widens, and your skeleton is being eaten from the inside. If you are over 65 and you break a hip, there's a 20 to 30% chance you will die within 1 year. Not from the break, from the cascade that follows: immobilization, infection, blood clots, pneumonia, deconditioning. The bone that was supposed to protect you becomes the vulnerability that kills you. And the reason it breaks in the first place is not accident or bad luck. It is physics. Your bones are not fixed structures. They are dynamic composite materials. Collagen fibers for flexibility reinforced with hydroxyapatite crystals for hardness, like reinforced concrete. And like reinforced concrete, the material must be maintained. 2 types of cells govern this process. Osteoclasts dissolve old bone, breaking it down and releasing its minerals into the blood. Osteoblasts build new bone, laying down fresh collagen and mineralizing it. The balance between these two processes determines whether your skeleton is getting stronger or weaker. Roughly 10% of the skeleton is replaced annually, complete turnover every 7 to 10 years. Before about age 30 to 35, the osteoblasts outpace the osteoclasts. You are building more bone than you are losing. This is why peak bone density is reached around age 30. After that, the balance tips. The dissolving begins to outpace the building. Slowly at first, roughly 0.5% to 1% per year, but it compounds. The physics of bone remodeling is governed by Wolff's law, formulated by Julius Wolff in 1892. Bone remodels in response to the mechanical loads placed upon it. Stress stimulates osteoblast activity. Load-bearing exercise literally builds bone. Remove the load and bone is lost. Astronauts on the International Space Station lose roughly 1 to 2% of their bone density per month in microgravity. Without the constant gravitational load, the osteoclasts continue dissolving, but the osteoblasts have no stimulus to build. The skeleton dissolves itself. The same principle applies to sedentary aging on Earth. Less physical activity means less mechanical stimulus means accelerated bone loss. And after menopause in women, the decline in estrogen removes a major hormonal brake on osteoclast activity. Women can lose 20% of their bone density in the 5 to 7 years after menopause. The bones become porous, fragile, riddled with microscopic voids. This is osteoporosis. And the collagen in the bone, the flexible component of the composite, is subject to the same glycation cross-linking as collagen everywhere else. AGE crosslinks make the bone more brittle. The reinforcing fibers become rigid, and when the material is stressed instead of flexing, it snaps. Stiff collagen combined with reduced mineral density is a catastrophic combination. The bone is simultaneously weaker and more brittle, less material and worse material. Everything connects. The mitochondrial decay reduces the energy available for bone remodeling. The glycation crosslinks stiffen the collagen in the bone matrix. The water loss changes the mechanical properties of every tissue. The protein misfolding disrupts the cellular machinery that maintains the bone. Every system degrades every other system. Nothing fails in isolation. That is why aging does not feel like one thing breaking. It feels like everything loosening at once. Because it is. And this raises the question that matters most. If all of this is driven by the second law of thermodynamics, if entropy is the root cause, is the rate fixed? Is there a clock ticking at the same speed for everyone? Or can the rate be changed? Can the rate be changed? Is every human body on the same countdown? Or do some systems resist entropy longer than others? The answer is swimming in the dark waters off the coast of Greenland. The Greenland shark is the longest-lived vertebrate ever recorded. In 2016, a team of marine biologists used radiocarbon dating of the shark's eye lens, proteins laid down at birth and never replaced, making them a permanent timestamp, to estimate the ages of 28 specimens. The oldest was approximately 390 years old, born around 1620. That is not a typo. This animal was alive before Isaac Newton was born, before the invention of the steam engine, before the United States existed. It has been swimming, slowly, in near-freezing Arctic water for 4 centuries. Its metabolism is extraordinarily slow. Its body temperature is barely above the surrounding water, roughly -1 to 5 degrees Celsius. Chemical reactions, including the reactions that produce free radicals and glycation crosslinks, are temperature dependent. They follow the Arrhenius equation. The rate of a chemical reaction increases exponentially with temperature. At the Greenland shark's body temperature, the Maillard reaction that caramelizes your collagen is running at a tiny fraction of the rate it runs at your 37 degrees. The shark is not immune to entropy. It is just running the clock slower, much slower. The Greenland shark slows entropy with cold. But there is an animal that slows it without any temperature advantage at all. And that is far more interesting, because it means the rate can be changed from the inside. The naked mole rat is a small, nearly hairless, cold-blooded mammal that lives in underground colonies in East Africa. It is roughly the size of a mouse. Mice live 2 to 3 years. Naked mole rats live over 30. 10 times longer than their body size would predict. They almost never get cancer. In decades of laboratory observation, spontaneous tumors are vanishingly rare. Their protein quality control systems are unusually efficient. They produce a unique high molecular weight form of hyaluronic acid that appears to protect their tissues. Their cells are more resistant to oxidative stress. They are, by every biological measure, aging far slower than they should. These are not miracles, they are data points. They demonstrate that the rate at which entropy degrades a biological system is not fixed. It is variable. And this is the part that matters. Some of the variables are under your control. The most replicated longevity intervention in the history of biology is caloric restriction. Reduce an organism's caloric intake by 20 to 40% without malnutrition, and it lives longer. This has been demonstrated in yeast, worms, flies, mice, rats, and primates. In rodents, the lifespan extension is typically 20 to 50%. A mouse that would normally live 2 years lives nearly 3. The effect is enormous and it is consistent across species. The mechanism is not fully understood, but the thermodynamic logic is clear. Less food means less metabolic activity. Less metabolic activity means fewer electrons passing through the mitochondrial transport chain. Fewer electrons means fewer free radical leaks. Fewer free radicals means less oxidative damage. Less damage means less work for the repair systems. The Maxwell's demons have less to sort. The pattern degrades slower. But caloric restriction also activates specific molecular pathways. Sirtuins, a family of proteins involved in DNA repair and gene regulation, are upregulated. AMPK, an energy-sensing enzyme, is activated, triggering autophagy, the cell's self-cleaning process, in which damaged organelles and misfolded proteins are digested and recycled. Caloric restriction does not just reduce damage, it actively enhances the repair systems. It gives the demons more capacity while reducing their workload. Both sides of the equation shift in favor of maintaining the pattern. Exercise does something different but equally important. Sustained physical activity does not appear to extend maximum lifespan. The longest lived humans are not all athletes, but it dramatically compresses morbidity. It delays the onset of age-related disease. Resistance training preserves muscle mass against sarcopenia. Weight-bearing exercise stimulates bone remodeling through Wolff's law, directly counteracting the dissolution that leads to osteoporosis. Aerobic exercise improves mitochondrial function. It increases the number of mitochondria per cell, improves the efficiency of the electron transport chain, and reduces the leak rate that produces free radicals. Exercise also upregulates autophagy, enhancing the cell's ability to clear damaged proteins and organelles. The physics of exercise is the physics of mechanical signaling. When you load a bone, the stress deforms the mineral matrix at the microscale, and mechanosensory cells called osteocytes detect the deformation and signal osteoblasts to build. When you contract a muscle repeatedly against resistance, the mechanical strain activates signaling pathways that promote mitochondrial biogenesis, the creation of new mitochondria. You are not just burning calories, you are sending physical signals that instruct your cells to maintain themselves. You are giving orders to the Maxwell's demons. Caloric restriction and exercise are interventions you can start today, but there are others, still experimental, that target the machinery of aging directly. Rapamycin, a drug originally isolated from a soil bacterium on Easter Island, inhibits a protein called mTOR, the mechanistic target of rapamycin, which is a central regulator of cell growth and metabolism. When mTOR is inhibited, cells shift from growth mode to maintenance mode. They slow down proliferation and increase autophagy. It tells the cell to stop growing and start maintaining, to shift resources from building to repair, from expansion to preservation of the pattern. In mice, rapamycin extends lifespan by roughly 10 to 25%, even when administered late in life. It is the most promising pharmacological longevity intervention discovered to date. Senolytics are a newer class of drugs designed to selectively destroy senescent cells, the cells that have passed their Hayflick limit, stopped dividing, and begun secreting the inflammatory cocktail that damages their neighbors. In animal models, clearing senescent cells improves tissue function, reduces chronic inflammation, and extends health span. The first human clinical trials began in the early 2020s. The results are preliminary, but the principle is sound. Remove the cells that have become sources of entropy rather than resistors of it. But here is what matters. None of these interventions, caloric restriction, exercise, rapamycin, senolytics, or anything else currently known, defeats the second law. They modulate the rate, they shift the curve, they buy time. Some of them buy significant time, but they do not escape the fundamental constraint, because the second law does not negotiate, and it sets a ceiling that no intervention discovered so far has broken through. Even if you eliminated every disease, every cancer, every heart attack, every stroke, every infection, you would still age. You would still accumulate stochastic molecular damage that no repair system can fully prevent. The copying errors, the quantum tunneling events, the glycation crosslinks, the oxidative hits, the telomere shortening. All of it continues regardless of how healthy you are, because all of it is driven by physics, not by pathology. The theoretical maximum human lifespan is debated, but the data converge on a range. The longest confirmed human lifespan is 122 years. Jean Calment of France, who died in 1997. Supercentenarian data, people who live past 110, shows that past that age, your odds of surviving any given year are roughly a coin flip, and the coin does not get more favorable. You do not become more robust at 115. You are simply surviving repeated flips. Mathematical models based on the accumulation rate of molecular damage suggest a hard limit somewhere between 120 and 150 years. Not because of any single failure, but because the probability of all systems remaining functional simultaneously approaches zero. Every tissue, every organ, every repair pathway would need to hold, and the compounding probability of all of them holding decreases exponentially with time. Past 150 years, the pattern cannot sustain itself. The information is too degraded. The demons cannot keep up. That is the thermodynamic limit, not a wall. A probability curve that approaches zero and never quite reaches it. In principle, someone could live to 200. In practice, the odds are so small that it has never happened and almost certainly never will, barring interventions that fundamentally alter the physics rather than modulating the biology. But the numbers are not the whole story. And there is one more thing worth understanding, not about how you age, but about what you are. Your atoms were forged in stars. The hydrogen in your body, roughly 10% of you by mass, is primordial. It was created in the first 3 minutes after the Big Bang 13.8 billion years ago. It has existed since the beginning of the universe. It was there before there were stars, before there were galaxies, before there were planets, before there was life. The heavier elements, the carbon, the nitrogen, the oxygen, the phosphorus, the sulfur, the calcium, the iron, were manufactured inside stars. Carbon is produced in the triple alpha process in the cores of red giant stars. Oxygen is made by the fusion of carbon and helium. Iron is the end of the line for stellar fusion, the most stable nucleus, the element beyond which fusion consumes energy rather than releasing it. Everything heavier than iron, the iodine in your thyroid, the zinc in your enzymes, the selenium in your antioxidants, was forged in the supernova explosions that ended those stars' lives. Scattered into space, drifting in interstellar gas clouds for millions of years, swept up by the gravitational collapse that formed the Sun and the Earth 4.6 billion years ago, incorporated into the crust, the oceans, the atmosphere, absorbed by organisms, passed from organism to organism for billions of years. And now, temporarily, part of you. The atoms do not age. The carbon in your collagen is the same carbon it was when it was fused inside a star 8 billion years ago. The iron in your hemoglobin is the same iron it was when it was ejected from a supernova before the sun existed. The atoms will be here long after you are gone. They will be part of the soil, the water, the air. They will be incorporated into plants and animals and other people. The atoms that make up your brain right now were once part of ancient oceans, of volcanic rock, of dinosaurs, of trees, of countless organisms that lived and died and returned their borrowed atoms to the earth. You are not permanent. The atoms are permanent. What you are, the thing that is aging, the thing that will eventually stop, is the pattern, the arrangement, the information. The spectacularly improbable, thermodynamically unstable, entropy-defying organization of 7 octillion atoms into a conscious being that can look at the stars it was made from and understand what they are. That pattern is fighting the second law of thermodynamics at this moment. Inside every one of your cells, molecular machines are copying DNA, folding proteins, pumping ions, generating energy, repairing damage, maintaining the structure that is you. They have been doing this since you were a single cell. They will continue doing it until they cannot. And when they stop, when the last repair is made and the last demon goes quiet and the pattern finally dissolves, the atoms will not notice. They will continue on exactly as they were into whatever comes next. Indifferent, unchanged, eternal. You are made of dead stars. You are held together by information. You are being slowly dissolved by the most fundamental law in the universe. And for a brief, improbable, unrepeatable moment in the history of matter, the atoms are aware of this. That is not a small thing. That might be the only thing.